We want to welcome you all here to a session on modern-day map making. I am David Agus. I am a professor of medicine and engineering at the University of Southern California. I'm a cancer doctor, and I work with new technologies to try to develop ways to better understand the complex system that's us as a human and to bring new treatments to cancer based on that.
And I'm Danny Hillis. I'm an inventor and a technologist. I started out in computers and made some of the fastest computers in the world, which turns out to be a great way to learn about everything else. If you make a very fast computer, then lots of people call you up and say, hey, can you help me with this problem? Can you help with that? Which got me introduced to a lot of different fields of science. And so I have a company that invents things for people who have a science or technology problem that they can't seem to figure out how to solve. And this is a little bit of an introduction to how David and I got together, which is mostly we work for people that we've solved problems for before. So we work for big companies. But there are always a few people calling us up with some new problem. And usually, sometimes they're impossible problems, sometimes they're crackpots. So, we tend to put that as a sort of a list to call back one of these days. And I kept getting this call from a cancer doctor who, wanted us to measure proteins or something that, and I kind of heard of him and thought, I should call back, but this is a really hard problem. He probably doesn't understand how hard it is. And then one day I came in and I had messages on my desk from John Doerr, Bill Berkman, and Al Gore saying, call David Agus back. So I did, and he came to me with a really interesting problem, which is essentially a mapping problem. But she said, look, I treat cancer every day without actually being able to tell what's going on inside the body in the signaling between the cells. And that's what I really need to know to treat it correctly. And I just don't have a technology to measure that. Would you work with me to develop a technology to do that? And I gave him a long speech about how hard it was, and it turns out he knew all the reasons and had understood the difficulties of it. And so we got together and worked on a project which was part medicine and biology, but part a physics project, and part a computation project, because once you measured all of these things, you need to understand them and make sense of them. So that's how David and I started working together.
How we started dating.
And that collaboration has led to a lot of interesting other things, but including progress in the original thing that we've started on. And I can say today we're now starting, we can now measure the proteins in the body and start to map them, and I'm sure we'll talk about that now.
That was a decade ago, and in that last decade, we've spun off technologies. We've been given significant resources from the National Cancer Institute to try to understand cancer better. And what we thought today is that we would go through some of these technologies that create this modern-day map making, allow us to really peer inside each of us and understand the conversation that our body's happening on a daily basis. That conversation's been going on since we were born, yet we've never listened. And we now have the technology for the first time to actually listen in and peer in on that conversation. And so in the physics world, they call things of scales. In the biology world, we're learning to do the same. We've got the DNA, we've got the protein, and then we've got the other parts of us. So we want to talk about three scales initially. One is DNA, the other is protein. Remember, DNA makes RNA, and then proteins are what allow cells to talk to each other and that conversation to happen. And then we want to talk about something from without, not from within. It's called the microbiome. It's the fact that we have tenfold more bacteria in each of us than we do human cells in the body, and how they affect that complex system that's us. Well, let's first start off with DNA, because it's gotten so much attention in the media over the last 2 decades. Right? A decade and a half ago, we saw the front pages of the New York Times saying, human genome is sequenced, we're done. And yet, we haven't come that far since it happened. So each of us have 3 billion letters in our code. And this code is what allows us to be human. The differences in that code distinguish each of us from each other. And they're rather small numbers of differences. In those 3 billion letters, there's several million differences in each of us. But from them, we can start to learn.
Even further, there's almost very small differences between you and animals. And most of your genome is the same as a chimpanzee. Actually, most of
Are you insulting them?
a lot of it is no. Well, a lot of it is the same even with a grain of wheat or with yeast. What percentage do we share with yeast?
Oh, over 80%.
Yeah. So life, most of your genome is how to be alive.
So you wonder why you have an affinity for beer.
So it's instructions about how to be alive, and then a little bit of it is variations about how to be different than every other kind of living organism.
And you know, we've seen wins, right? We've seen wins where certain genetic sequences, if you have them, can tell us that you're more likely to get disease X or disease Y. I can look at your DNA and tell you your lifetime risk of getting Alzheimer's disease, of getting heart disease. At the same time, if someone's unfortunate to be diagnosed with cancer, I can look inside the genes of the cancer and start to say, how is it different from the normal body? Is there something I can target? Is there an on switch there? And there have been many over the last decade, and it's yielded some minor advances in cancer. And so, Danny, as a physicist, and you know what I'm going to ask here, but I can target these DNA changes in cancer, and I can buy a couple months of extra life and quality life for patients, but why aren't I fixing the problem?
Yeah, so the truth of the matter is that people describe your DNA as a blueprint, but actually it's not a blueprint at all. It's a list of ingredients. And so you don't really know anymore. Once you have that list of ingredients, you might know something. But imagine if I gave you the list of ingredients that a restaurant uses. Can you tell if it's a good restaurant or not? Can you tell if it's a healthy restaurant or not? Well, maybe if it is a French restaurant and it doesn't have any butter, you could say, well, there's something wrong with it. And there are some diseases that are like that, where we're missing an ingredient. Cystic fibrosis is like that, for instance, where you're kind of missing a critical ingredient. But almost all diseases are not in the ingredients list, because actually healthy people have the same genome as sick people. The same person when they're healthy has the same genome as when they're sick. So the genome isn't really the thing that tells you if you're healthy or sick. In cancer, yes, you have some cells that maybe get some mutations, but you know, all the DNA can really tell you is, do you have predispositions to do certain things? And it's actually kind of weak evidence. It's surprisingly weak evidence. For instance, if I knew your genome, right now, I actually couldn't even reliably tell you the color of your eyes in all cases. I couldn't tell you how high you were if I knew it. Huh. So we don't really understand those, we really don't understand the meaning of those letters enough for them to be that useful in treating disease and
I'll take it one step further. Yeah. It's pretty cool, is that there's a kind of leukemia called chronic myelogenous leukemia where 2 chromosomes break apart and they join together. There's actually a drug called Gleevec that many of you probably heard of that can reverse this leukemia. Well, if I walk through the streets of New York City today or Aspen and I draw blood on 100 people, 3 of those 100 will have the chromosomal changes of leukemia, yet they don't have leukemia. So you don't just need the DNA, you need the environment to actually be receptive. It's like if you want to start a fire in the woods and you light a match, you don't just need a match, you need the wood to be dry. If it rained the night before and drop it, nothing happens.
So here's an example of how we were really wrong about what we thought was going to happen with mapping the genome. So that's the first big map we've made. So people assumed that there was a gene for almost everything. So there would be a gene for eye color, there would be a gene for height, and that was the way the biology textbooks were written. And so the assumption was that once we had the list of all the genes, what we could do is we could just take out a gene and see what broke, and then we would know what that gene was for. But guess what happens when you take out a gene almost all the time? Absolutely nothing. So the way that your body interprets the genes is somehow clever enough that it corrects for problems. So even if you deliberately introduce a problem in the genome, almost never does it have any effect. And so our body is this fabulous system for sort of taking this book with a lot of typographical errors and making sense of it and creating a healthy individual. And so the correspondence between the genome and health is a very, very complicated thing that I think we're only beginning to appreciate how complicated it is. And so in between the genome and health are a bunch of other complicated things that we're going to have to map, which are some of these other things that David mentioned. We're going to have to start understanding what are the proteins that get built, which are the actual signaling in the body, how your body talks to itself to regulate, to grow, to fight off disease. And then also we've begun to realize that it's not just your body, it's the whole ecosystem of not only your DNA, but all of the things that are living inside you, which, as David said, there's more DNA of other species inside of you than there is of you, which is really, I mean, how new is that information? Where did that?
It's all come in the last 2-3 years. For the first time ever, we started to understand the microbiome. We're going to talk about that, but I want to do a timeout now and do something very unorthodox. You know, I've been coming to Aspen now for, I think, about 8 years, and it's had an enormous influence on me, and all of you have an influence on me. But about 4 or 5 years ago, Aspen put me together on stage, this same room, with one of the greatest scientists of our time, a guy named Murray Gell-Mann. Murray Gell-Mann is one of the great physicists, identified the quark, string theory, founded the Santa Fe Institute, and has had a profound influence on me and how I think about disease and much of what we're talking about today, and has had an important part in Danny and our relationship. And Murray just walked in the room, and so we all want to give thanks to Murray and what he's contributed.
Murray, in fact, played an important part in my intellectual development because he was somebody that you could always go to with any question, because Murray knows pretty much about everything. You can go to him with a question of linguistics or physics, of course, philosophy. And in fact, Murray has a habit of when he meets people, he explains to them how their name should actually be pronounced. And can explain exactly the root of their name and, you know, the language history of their name and so on. But my favorite moment with Murray, because it just struck me, the truth of it is, once I had a very obscure question in quantum mechanics that I had sort of gotten into an argument with a colleague. And I was like, okay, well, let's go ask Murray Gell-Mann. And so we went and we posed the question to him. And Murray sat politely and he listened to it. And then he started giving an answer. And in the course of his answer, he made something that I thought was a bit of a speculation. And so I said, well, Murray, I mean, we don't really know that. And he looked at me slightly offended and he said, well, you're right, we don't know that. In fact, you know, the field doesn't know that. He says, but I know that.
That's Murray.
And he was right, he did. So it was really, it's been an extraordinary privilege to be able to work with Murray. And in fact, Murray has worked with us on this cancer project that we've done together, trying to understand the development of cancer in mice.
No, it's been a true privilege to work by his side. He and I published a paper about a year ago. It was called A Physicist and a Cancer Doctor Walk into a Bar, ... And it was the notion that in his field, everyone in the world does theories, right? The physicist came up with a theory and someone can knock it down or build it up, but the theories brought in all the experiments and put them into one way of thinking. In my field, nobody does theories. And so Murray and I wrote that paper, and Murray had done his edits, And he called me up because I'm ready to send the edits. How does the fax machine work? The smartest guy in the world, and we couldn't get that fax machine to work. But it truly has been eye-opening for myself, for Danny, for all of our team, to have Murray give his insights from the physics world and cross-discipline into the cancer and the biology space. And we have to thank the Aspen Institute for that, because it's had an enormous influence on what we've done, and obviously Aspen has had an enormous influence on Murray.
But actually this point that you just made about theory, I think is the essential thing to understand about genes and genomics, which is in physics, there have been many examples where people have made a theory of something and then gone to a microscope or a particle accelerator or a telescope and discovered the thing that they theorized. And for example, Murray theorized the existence of quarks, and it was entirely on mathematical grounds that he believed that they should exist. And it was many, many years before they were observed. And so that's a very common sequence in physics, to come up with a concept and then much later find it and observe it. And in some sense, in biology, I think that's only happened once. And we can argue about this, but I think it's only dramatically happened once, which is the idea of a gene.
Well, the idea of DNA at the time.
Well,
yeah.
but DNA wasn't, the gene was hypothesized. Okay. So gene was hypothesized long before, Mendel hypothesized the gene long before we knew about DNA. And the idea that there was a particle of inheritance. So that, and had the notion that in fact everybody had sort of two of these particles. You could kind of just tell that by the way that things crossed and hybridized and bred and so on. So biologists knew that there was some particle of inheritance. And then that was why what Watson and Crick did with DNA was so amazing, because they discovered the particle of inheritance. They discovered that how that information was encoded in DNA. But the theoretical idea of inheritance was that you would have a gene that determined eye color, for example, a gene that determined height. And since these things were obviously heritable, the assumption
the P
was
screwed us up.
Yeah, well, the genes, that idea of genes did screw us up. So that's why we were so optimistic that when we mapped the human genome, we would kind of understand everything. We'd be able to see, well, where's the gene for this? Where's the gene for that? And the assumption was that, you know, we would be able to tell, for instance, are you going to get a neurodegenerative disease? When are you going to do it? That ought to be in the genome. And what we discovered when we mapped the genome is, in fact, this relationship between the genes and the phenotype, who you were, was much more complicated than expected, and was much more about an interplay of environment and genes.
Well, there are
so
other experiments that kind of played
else.
in. You know, in the 1920s, an amazing experiment was done where they took people with a significant cut on their leg. Half of them, they took a piece of bread and they dipped it in water and wrapped it around the leg. And half of them, they left open to the air. The people who had the bread on their leg healed twice as fast. And it started something called germ theory. And germ theory, again, said, as soon as you look into the microscope, identify what you're up against, just like if there's one gene, you know how to fix it. And while it's right for bacteria, that's not right for heart disease, cancer, Alzheimer's, any other human disease. Right. Because it really is much more complex than that.
Yeah, so there's a, you're right. So that's another area
you said
where
I'm right? Oh my gosh.
Well,
this is shocking.
Well, I think you did pick another example where there was a theoretical concept which existed before we actually found the particle. So you're right, the germ is another one.
Yes.
But again, the germ is one that we overgeneralized. And so medicine had fantastic success at killing germs, and antibiotics being the obvious example. But also public health, too, of keeping the germs out of the water supply, for example. And so that was really the germ theory made medicine go from essentially voodoo to something that, you know, actually had a theory and a practice that could really cure people and really prevent people from getting disease. But the problem is that medicine then got into the habit of treating disease as if it was caused by an invading germ. So the paradigm for medicine became identify the disease by the character of the invader. So is this cholera? Is this typhoid? And then kill the invader. So kill the disease, or cut out the disease. That was the basic paradigm of kill the disease. The problem is, if you take a disease like cancer, the disease is not something coming from the outside. It's not, in fact, it's not a thing at all. There's not a, we tend to identify cancer with the tumor, for example, but the tumor is really just a symptom of the cancer. Cancer is actually something you're doing. In fact, it should probably be a verb, you're cancering. And so the paradigm of going in there and killing the cancer is just the wrong idea. And I mean, it's funny, I'm explaining to you things that I've understood from working with David a long time.
Well, let me tell you another story, and maybe that'll start a new end to the conversation. And so this is a paper that was published at a Cleveland clinic several weeks ago. And you've all heard of the concept of stomach stapling or gastric sleeves. And there was a notion, rather simplistic, that if we make the stomach smaller, someone who's obese, someone who's large, will eat less, lose calories, and get skinny. So they did a clinical trial, and they took people who were obese. Turns out when you're obese, you also have diabetes, right? People are very large, almost uniformly, over 90% have diabetes. So they went through this surgery, and then about a month later, in 95% of them, diabetes went away. 3 months later, they started to lose weight. So diabetes went away before they lost weight. And when they look closer, what happened is that when you change the architecture of the stomach, it's not that you ate less food, it's that you changed the bacteria that lived in the stomach and the GI tract. And by changing this bacteria, you change what you absorbed. And it went off an experiment that happened 2 months before that, where they took bacteria from an enormous human and they put it into a skinny mouse. 5 days later, the mouse got bigger. They took bacteria from a skinny human and they put it into a fat mouse. 5 days later, the mouse got skinnier. And so going forward, we're not going to do this rather barbaric concept of stapling your stomach at a cost of $27,000. We're going to change the bacteria in the GI tract, which will actually change the weight of the individual and what they absorb from food. Turns out that if you look at the risk of breast or prostate cancer in China, they're 1/10 that in the United States, and look at deaths. As someone from China moves to the United States, after a decade, their rate starts to approach ours. And if you ask people, it's the Burger King and McDonald's, turns out it's not. It's that they adopt our microbiome, our bacteria, which changes risk for disease. And by the way, you're not scot-free in China. They've got gastric cancer, lung cancer more than we do. So it all evens out in the end. But the notion that this complex system that's you and I can be modulated or changed by the bacteria in us is very powerful. And there are 21,000 different bacteria in each of us. So Danny, how do you understand something with 21,000 different species? How can we modulate that? What are we talking about?
So it's a special case of the more general question of how do you understand something which is literally hundreds of thousands of different kinds of communicating things, some of which are these bacteria, but some of which are also the organs of our body, the cells within our body, our immune system. And so how do we understand, if you look at it as a system, our body is a kind of society. There's politics going on, there are revolutions going on, it is more like a culture, more like a city than it is, like a machine. And so how do you understand the complex interactions of all of these things? And obviously what you have to do is you have to listen in on the communications between them. And so you have to be the kind of the NSA listening for the chatter.
So NSA is good, you're saying?
Well, that's a whole other discussion.
Okay.
But listening in, you can begin to understand that, hey, a revolution is brewing in your colon. And so how does the communication happen? It's not genes. The genes pretty much stay the same. The way that the communication happens is deliberate signaling by these things called cytokines, which are proteins. And actually, in general, it's proteins are carrying the information. Those are the things that the DNA makes. And so those are the language that the body communicates with itself in. It's the language that cells communicate with each other. It's the language that the microbiome communicates with the cells, and so on. So what we needed was the ability to listen into that. And that was what David's phone call to me was about. So we have to be able to listen in on that conversation in order to really understand what's going on with the disease. Because right now, what we understand is where does the symptom show? So if the symptom shows up in the lung, we say it's lung cancer. If the system shows up in the breast, we say it's breast cancer. But that really doesn't say very much about what's going on. It may just be coincidence that it showed up in the lung versus the
if I took out a liver of somebody with colon cancer metastatic to the liver, and I showed it to a surgeon, they'd say, Agus, that's colon cancer metastatic to the liver. I showed it to a pathologist, they would look under the microscope and say, hey, colon cancer metastatic to the liver, no doubt about it. And so it looks a certain way. If I looked at the genes, there could be 1,000 different mutations in DNA to get there. So it all looks exactly the same way, and yet the DNA can be radically different. You know, it's akin to what you were talking about before, the ingredient list. Well, proteomics, looking at these proteins, are what the food tastes like. And so it's a very different way of approaching it. You know, in 1976, if you, your friend, your spouse thought they were pregnant, we took a tube of blood and we injected a rabbit. And then 5 days later, we sacrificed the rabbit. If the rabbit's ovaries were enlarged, you were pregnant. That was the state of the art in 1976. Well, in 1977, along comes the first protein test where they looked at one protein, HCG, and they could say for $9 whether you were pregnant or not. It radically, obviously, changed the lives of rabbits, but it also changed the lives of, newborn babies and of mothers, because we could know what was going on. Well, Danny just described looking at the entire conversation in the body.
Which is literally 100,000 different proteins. And some of those are proteins, human proteins, some of those are from the microbiome, and we're looking at the levels of those changing. And so in that is the information that's much more useful for telling it's a much more useful idea of what kind of cancer do you have. Instead of saying, well, you have the kind of cancer that causes a lump in this part of your body, what you'd really like is you have the kind of cancer that responds to this drug. That would be the really interesting thing.
Or along your, akin to cancer being a verb, is I want to figure out how to change you from a cancer state to a health state. My job as a cancer doctor is not to shrink your cancer by 50%, which we call a win in our area, but it's to change you so the cancer didn't want to grow. There's a clinical trial done about 15 years ago that at the time people said was unconscionable, because they took women who were premenopausal with breast cancer, the worst there is, young women, and they made them, it was unconscionable because they made them come in once a month after their treatment to the hospital to get an infusion for several hours. And they said, let them be home with their family because that's what counts. And this may be their last months at home. Don't make them come in. But they gave a drug that made them build bone, make their bones stronger. They reduced recurrence of the breast cancer by over 40%. Why? Because breast cancer metastasizes to bone. So if you change the soil, the seed doesn't grow. So one of the greatest survival advantages in breast cancer from a drug that didn't touch the cancer, it changed the system so the cancer didn't want to grow. And so Danny's been describing a way, for the first time, we can actually see that system.
So for instance, that's a great example of getting out of the paradigm of killing the cancer and into the paradigm of adjusting the body toward health. So by making the bones stronger, it made it harder for the cancer to invade the bone, and therefore it made the patients much less likely to have recurrence of the cancer. And so that's more the direction we want to go. But to go in that direction, we have to have a better classification. And we have to have a better signature. So we've just recently gotten the technology now to measure this. And so we're just starting to look. But for example, it looks very likely in what we're doing right now that we can see colon cancer as it starts to form. Well, why is that interesting? It's interesting because if you catch colon cancer in the very, very early stages, it's completely treatable. In fact, if you catch it, we can even probably see it before it turns into cancer while it's still at a polyp. And so you can get a colonoscopy, get it removed, and never have it progress to colon cancer. So if this technique works, nobody ever again needs to get colon cancer. I mean, that is
But even
incredible.
more than that, I mean, obviously most of us have had colonoscopy. Most of us don't have a polyp, which means we didn't need the colonoscopy. So wouldn't it be neat if you could draw a tube of blood and say, hey, you need a colonoscopy because you have a polyp and you don't, to come back in a couple of years. That will radically change medicine. Most people never get a colonoscopy because it ain't fun. And colon cancer is one of the top 3 killers of all cancers because of that. And we can totally eliminate a disease if we do this right.
So that's an example of something, if we could generalize that. So right now, you can correct my percentages on this, but I'd say 95% of medicine is about treating sick people. And maybe 5% is about keeping healthy people healthy. But notice this is something new. So medicine in the future is going to be not so much concerned with either this, well, it's going to be concerned with the people that are in that middle state. They're not totally healthy, but they're not sick yet. They're in that stage where something is starting to go wrong, but it's not gone wrong enough that they're showing any symptom yet. And that's the time you want to treat it. You want to treat people before they're sick.
Well, Danny, what is health? I mean, you keep saying that word, let's keep you healthy. I don't know what that is.
Yeah, it's kind of, I don't know what being alive is exactly, but there's a big difference between being alive and dead.
Okay, so is it the absence of disease?
So I think it is, I mean, it's an interesting question. I mean, obviously, I can more point to the negatives and say, you know, when you're diseased in a way that's interfering with your activities, and causing you to die earlier, then that's not healthy for sure. But
okay.
You know, what is, so to me, you could imagine, and maybe this is never true, somebody who has absolutely nothing, raw, everything is under control in the body. So your body is dealing with all your problems.
Right, but you may still have
successfully dealing with all your problems.
You may still have a cancer that's this big, but it's not big enough to cause a problem.
Well,
and
certainly.
I certainly wouldn't think they're healthy.
Well, I don't know. I mean, so I would call people healthy if their body is successfully dealing with it.
So the day before you have a heart attack, you're healthy?
No, well, that's not, you're not successfully dealing with it if you're heading toward a heart attack. But certainly, I mean, probably most of us have small cancers that our body has successfully encapsulated, shut off the supply to, and they will never cause us any problem. So that, to me, doesn't prevent you from being healthy if your body has successfully stopped a problem. I mean, first of all, I want to make sure that's true.
Well, I'd view it a little bit differently in saying I'm not sure it's an active process. It may be just passive, that the molecular changes of cancer can happen, and it doesn't grow in that environment, or your body isn't right to allow the cancer to grow or to do its thing. So I'm not sure, we don't have evidence that it's actually an active process of containment.
Okay.
It may be passive.
But you would agree that we get cancer cells all the time when we deal with cancers.
We have the molecular changes that are akin to cancer. But to me,
and
cancer is a process that hasn't become a cancer yet. It's got the DNA changes that are cancer, yes.
Okay, so wouldn't you say that somebody's healthy if they're doing that successfully? I mean, that's a piece of being healthy. So
yeah, but they could
just because you
could be 400 pounds and they can't play with their grandkids. Are they still healthy?
Well, okay.
But they don't have your cancer.
No, they're not. Okay, so if it's interfering with the things you want to do, it's not. That was one of my definitions. If it's shortening your life or interfering with the things you want to do.
Again, we're getting a little off tangent, but I think the notion of developing a way to measure the body to say, are you in a state of health or a state of disease is going to be very powerful. Yeah. And we have the technology is starting to happen for the first time we can actually see that.
So I think we agree that probably we're going to go to a world where some interval, say once a month or something like that, you prick your finger, you put a little blood drop on a piece of paper, and you mail it off, and somebody analyzes that and says, oh, you are going in a bad direction if you don't do something different.
Let me tell you a different story
okay.
to kind of put things in a different perspective. 1953, an amazing paper came out in the New York Academy of Sciences. by this remarkable woman in New York. And nobody paid attention to the paper, and they actually called her Vampire Dracula and all these crazy names. What she did, kind of wild, is she took two mice, an old mouse and a young mouse, anesthetized them for 3 weeks, tied their skin together to allow the blood supplies to mix. And what happened is the blood from the young mouse went to the old mouse and vice versa. She then looked, and turned out the old mouse, the brain all of a sudden had new connections. The muscles in that old mouse got stronger. The heart beat better in the old mouse, and the young mouse actually aged. Some of the hair that had turned white on the old mouse reversed and was brown again. In her paper, she was a very strong political bias. She would say over and over, the reason we're not affecting aging is we're spending too much on war. And she added all these political things into a science paper, which I've never seen before, but people ignored it. Well, 4 weeks ago, there were 3 separate papers that came out in the top science journals out of Harvard, UCSF, and Stanford, and they all showed the same thing. They showed that if you took some proteins from the blood of a young mouse, and they actually identified it down to 1 protein, what would happen is it would turn back on the stem cells in the adult mouse. Remember, evolution selects out for who has good children. It doesn't select out for who lives until 90 or 100. And so you don't need those stem cells once you're past childbearing years, according to evolution. And so what this did was it turned on them, and it actually reversed aging in these mice. And the clinical trials in humans started a week and a half ago. And so, when we start to look at this, an observation took 60 years from that initial observation for someone to actually jump on it and explain it and to change that system. Danny and I have the conversation over and over again how a kid, if they get cancer, obviously a horrible, unfortunate thing, but it's rare, several thousand cases a year in the United States, but over 90% of those kids have a curable cancer. 90% of pediatric cancers are curable. In an adult, once you turn 25, a resiliency switch gets turned on. And that cancer, no matter what you do, it gets stronger. Remember, this is one of the few diseases that every time you treat it, gets more aggressive and stronger. So that switch, something's going on.
So you said something really quickly, which I want to say more about. You went over something, which is the comment about evolution. And I think it's worth explaining. Which is...
Oh, you're not anti-evolution.
Oh, no. Oh. I agree with you on that. I just think you went by it so quickly.
Got it.
People may have missed it. But evolution basically selected for our ability to copy our genes and have children. And so really, evolution is only interested in our ability to do that. And so once we've past childbearing age, evolution, there's really no selection pressure in evolution to sort of keep us alive. Maybe a little bit to take care of the children, but it's pretty weak. So in some sense, we're past the point where evolution is really trying to fix the problem, which is why cancer is, in fact,
we
a
being
disease.
The people of our age.
Yeah.
There are younger kids in the audience too, yes.
We humans,
yes.
That's right. But so, that's right. So we humans, basically, once we're past childbearing age, we're in kind of uncharted territory as far as evolution is concerned. And so we're kind of on our own. And in fact, cancer tends to be a disease of older people. And as David points out, even when it does occur in younger people, it's much more curable. And so in some sense, what we're doing is now that we understand the genes, now that we can map the genes, we're taking control of a thing that evolution used to have control of. So today, we're actually getting to the point where we don't just have to accept the genes we have, we're actually getting to the point where we can change the genes that we have. We can write new genes. We can put new gene into either, we can do it with an adult, or we can do it with a child. We can have our children born with, the traits that we know are going to be most useful for them fighting off disease, for example. And so that creates tremendous new possibilities, tremendous new moral dilemmas. But we're in a new world where we're deliberately writing our genes instead of just kind of being stuck with them, instead of depending on evolution to pick them for us.
And obviously that whole world is changing. You know, in the 1990s, there was a team that was given $25 million from the government to identify the faulty gene for breast cancer. They actually did it in the Mormons because Mormons wanted to be buried in the church and they couldn't fit. So they took hair from each of the Mormons along with their history and they were in the church. And they went back to that hair, that DNA, and they identified the gene. It was licensed to a company that went bankrupt and another company bought the assets and sold that test for $5,000 or $6,000. And so they basically, you know, this is a test that would cost $50 to run in a lab. And so they basically held the genes hostage. And the Supreme Court, about 7, 8 months ago, announced that you can no longer patent a gene in our country. So they've democratized DNA. And they've really allowed that field, hopefully, to flourish because each of us now have the right. It's hard to imagine we didn't before, but we have the right to look at our own DNA. And as we talk about this, I want to switch gears totally and talk about a topic that's come up a lot these last few days at the conference, that how it's been presented isn't exactly how I would have presented it, and that is the topic of genetically modified food. And so as we talk about genetically modifying you and I and how that's possible, let's just talk a little bit about what we call GMO, genetically modified organism. It's the notion that instead of what Mendel did is, you know, breed pea plants to try to get a particular trait and keep doing that over long periods of time, we can quickly, to circumvent the time, put in a gene for a trait we want.
Yeah, so think of, we talk about genetically modified organisms as if they were the only ones that were modified. But in fact, the way we used to modify organisms, before we had genetically modified organisms, is we depended on radiation to randomly modify organisms. And then we took the randomly modified ones and then we took the ones we liked and we bred those traits and did it through a process of hybridization. And all the food we eat today, except for GMOs, is dependent on randomly modified organisms. So it's really a question of randomly modified organisms versus deliberately modified organisms. Now that's not to say that there aren't dangers with deliberately modifying organisms. But I think there's a sort of false idea that somehow one set is modified and one set is somehow natural. Almost none of the foods we eat today are natural. They've been genetically engineered by a process of breeding that depends on random modification from radiation.
It's interesting. Some of the foods, you know, that have an olive tree that's 100 years old certainly hasn't had that same degree of breeding and selection than corn, which is cycled every year.
Yeah, but pretty much anything that humans eat as a crop has been selected over many years. And certainly, even a completely natural thing that we've never used as a crop was randomly modified by the process of evolution.
And given that,
so it has a collection of genes. So
4 crops represent over 70% of the calories that the world eats. Yeah. That's not a difficult statement to make.
So what we can do with the gene is we can put one of these particles of inheritance. Now we can actually create it in the laboratory and put it in, and then the future plants will inherit that. As opposed to before, we depended on radiation to naturally cause that mutant, and then future crops would inherit it. Now, the big danger of that, I mean, I think there are some real dangers. One, we can change things more quickly. So that old process took years and years, and so we got to see the consequences of the changes fold out a little bit more slowly. So there's some very good arguments about being careful about how we use GMOs. But once that has happened, if I took, let's say, a kernel of corn that I deliberately put in a gene, and a kernel of corn that just happened to get that same gene by the natural process of radiation, there's really absolutely no difference between those two things. And so that's why an awful lot of the arguments about GMO foods...
Well, I'm not sure that's a fair statement. I mean, you know, because again, there's no way that you're going to find in nature that you took a gene from a rabbit and put it into a corn. So,
well, that's actually not true. I mean, we do know that...
we do from viruses, but we don't from other mammals.
That's right.
We do know from genes hop.
We do know that genes hop.
Right. There's some nuances to it. But the problem I see, though, is that you're optimizing on one parameter, which is, in general, calories per acre.
Yeah. And so what you do with that ability to modify, I think that's where the discussion should be.
Right.
So for example, I think that one of the big uses has been to make corn pesticide resistant. I'm sorry, herbicide resistant. So that, and that's what you probably heard in the Monsanto discussion, that Roundup Ready crops. What that is Monsanto developed a line of corn and other crops which were very resistant to an herbicide called Roundup. That's their brand name for it. So that you can spray this herbicide on the crops and kill all the weeds, and the corn will keep going. And so when it first came out, it had a fantastic ecological effect of causing people to use far less herbicide to keep the weeds down in their crops. And so herbicide use went down. Seems great. Seems like they made a great ecological contribution. But one thing that has happened as a side effect of that, because they picked that sort of one thing to change, is that a new generation of weeds is beginning to become more and more resistant to that herbicide. And so evolution is kind of now working on the other side of it, and farmers are discovering that they have to use more and more herbicide to keep the crops down. Now, so we could go back and argue, was the benefit of that enough for that period of time, or they will move on and they'll put other genes inside. Was that a good use of GMO? I think that's a legitimate argument, and I can see arguments on both sides of that. But that's the kind of argument I think we should be having, not the fact that we deliberately modified this gene somehow makes it into a different thing. Now we could do, for instance, deliberate modifications to grow healthier corn with more vitamins. And would that be a bad thing? Well, there might be some reasons it's bad, but I think that now that we have the power to deliberately modify organisms, it seems to me like the discussion should be, how do we use that power? How, a thoughtful use of that power, rather than the discussion of, let's stay away from using that. It's a little bit superstitious, I think, to feel like somehow if we've deliberately changed something, we've made it into something different than normal life.
Well, we've gone all over the place in the last 30, 40 minutes, and what we want to do now is open it up to questions. And so, again, the conversation has been on multiple sort of aspects, and I hope you gained some insight to how we're viewing technology and how cross-discipline we can argue back and forth. But let's go to some questions. Yes, and I'll repeat the question. There's a microphone floating. We're going to, if you can catch the microphone, it'd be great.
Let's start with people near the microphone.
Yes.
For efficiency.
How are you, in fact, mapping these proteins, genes, the microbiome, and the other bodily processes that you're talking about? You've talked about how they interact, and you said that you were mapping them, but you haven't told us how you're mapping them, what technologies you use.
It's a great question. I think technology depends on what we're looking at. So DNA, there's been more and more technology to rapidly and a very low cost sequence, that is, get the letters A, C, T, G, and put them into order for the 3 billion pairs. And there are 5 or 10 different technologies out there that can do that. They all presumably give similar information. For the protein, we're taking a superconducting magnet, and based on the physical properties of the protein, we're separating them out and getting a picture of everything going on in the blood. The microbiome, the bacteria, we're using that DNA technology again, and Danny may be the first to argue that it's probably the wrong technology to characterize such a complex system.
Yeah, and this is an area of rapid development, and I mean, David and I have actually made some new instruments, but probably they're going to be obsolete in a couple of years. They'll be replaced by even better ones. And so once people start using the information, then the physicists come in and start inventing all kinds of new ways of measuring it. And so that's making rapid progress. With proteins, I think we just went over a line that really we could not consistently measure the proteins a couple of years ago. Now we can.
Well, let me ask you a question. Do we need, akin to what was asked, to actually map all of them? Or can we use what you in the physics world call coarse graining as ways to understand these systems better than actually mapping them?
Yeah, well, and of course, we don't know yet. So it's sort of like we've just got the first telescope working, and we're looking around at the stars, and we're discovering Jupiter. And so we're at that moment in proteomics. So for instance, we don't even know how many proteins there are. There are more proteins than there are genes, because your genes build proteins, but then the proteins modify each other and add things. And of course, you have the proteins from the genes of your microbiome. So, are there 100,000 proteins? Are there 500,000 proteins? Or we used to think that there were only 20,000 proteins, but we're seeing way more than that. So we're just beginning that mapping process with the proteins. So it's a fantastically exciting time in that field right now.
What do you believe is a correlation between eating GMO food and cancer?
I think, again, this is my opinion. This is not, there is no study showing that eating GMO food leads to cancer. That being said, is that there is no study showing that eating GMO food is equivalent to eating non-GMO food, because I'm not sure what to measure. And so GMO food has been optimized to not get bugs, not get infections, maybe to grow when there's less rain, but it was never been optimized to say how healthy is the person that eats it. And so as an engineer, you pick an optimization parameter for everything you do. When those optimization parameters don't take into account long-term health of the eating person, that worries me. Although, again, I'm not saying that there are studies that show it causes a problem, but it does significantly worry me.
But I think you skipped over something in that.
What?
Which is, it depends on how it's modified, obviously. So the fact that a food has been modified is not what controls whether it is healthy or causes cancer, or because it's how it has been modified. So I'm sure you could deliberately modify a food in a way that it was carcinogenic. And I don't know why somebody would do that, but you could deliberately modify a food in that direction. You could also deliberately modify a food to take out a known carcinogen. So one of those kinds of things would presumably increase rates of cancer, and one of them would presumably decrease. So I think that the fallacy is the fact that it's modified, I don't think is the determining thing. I think it's exactly what is the modification that was made. So I'm sure that there are bad modifications that can be made.
Yeah, but classically in nature, when things are modified, when you select out for the bigger tomato, it's not because of one gene. When we're modifying foods, we're actually giving large amounts of one thing, which in a complex system may portend...
Again, you're talking a how, though. You're saying a way that things... So things
yes.
So one of the things about GMO is very often larger modifications are made than are normally made. So you can say, well, that's more dangerous if you make a larger modification. And I think that's a legitimate argument. So, but there, I think what you're saying is, well, maybe we should only modify foods slowly rather than quickly. And I think there's a legitimate argument for that. But I don't think it really depends on whether we're deliberately modifying. If I can modify it quickly by exposing it to radiation, is that good? Is that better? So that's not a GMO food. If I could modify it by putting in a particle accelerator, I get something that's not a GMO food. It's a natural food, but it's been modified quickly. Well, so yeah, I think we should be more afraid of things that have been modified quickly, and we don't really.
Well, I think we just need to set up standards. How we look at how it affects health, how we look at how we report how it's been modified, where it's been modified,
yeah.
And we can start to follow them
yeah.
and have transparency and labeling, so we can understand what's going forward.
And that is definitely an area where I think that the people who are doing GMOs have really gone in a counterproductive direction by resisting labeling. And I think it works against them. It makes everybody suspicious that there's something wrong there. I think actually what would happen if you started labeling genetically modified foods is you would be shocked to discover that most of the things you eat in fact, are genetically
I think
modified.
what you're sensing is, I mean, we're afraid of throwing out the baby with the bathwater. We believe in science. We believe that genetically modifying will have a role. You cannot move the wheat fields of Nebraska to New York City with global climate change. But you need to do it ethically, responsibly, and with true science behind it, and that's what we're pushing for.
Right. And so it's like any other technology, any other powerful technology. It's not, technologies in themselves are not good or bad. If you take antibiotics, I mean, antibiotics are really great if you've got a sick child. It's probably a terrible idea to feed huge amounts of antibiotics to chickens, just to get the chicken productivity up. So there's bad ways of using technology, and there's good ways of using technology. So I think it would be much more productive if we could manage to shift the GMO discussion on how do we want to use it, rather than is it a good technology or bad technology.
Let's get more questions. We can kind of get off chicken productivity if you think that's... Yes?
Hi, yes. How close do you think we are to using either DNA sequencing or protein analysis or some combination of those to routine cancer screening?
Screening or treatment?
Screening.
Very close. We're all, we're
use it
already
now.
starting to do it.
That gene I told you about for breast cancer gene is, we use that routinely to screen for risk of breast cancer.
But I think you'd agree what we're doing now is...
Like, that would be used in combination with a mammogram?
No question about it. I mean, it's being used now, and it's being used now. People with a family history, insurance pays for it.
Right.
But you're right. In the future, it's going to be much more broadly as the costs come down.
Yeah, so now it's kind of a by exception thing. In special circumstances, we use it, or we look at, for instance, proteomics. We'll look at one protein, like PSA is a protein.
So it's for people who are at a higher risk.
Right, but it's because of our weakness. Is that that breast cancer gene is hundreds of thousands of letters. We know what a couple letter changes mean. But if I did you and you had no risk for breast cancer, and I showed that you weren't like the 10,000 women we tested before, I might say you have high risk. And we just had a case like that before. Is that a friend of mine called me up and said, I got told I had a genetic change in the breast cancer gene. And I actually was able to go into the data, because the database is held by a company and you have to pay to access it. I was able to go in and look and tell her that genetic change won't cause a problem, and she was ready to go and have surgery. So we need to learn more about what they mean and that it'll be commonplace, but it's still early. We still got the first telescope.
But I will, I'll make a prediction, and tell me if you think this is true, which is every time you go for your annual physical for the next decade, every time they will measure more proteins, they'll measure more biomarkers of the sorts that we're talking about today, and inform you more about your health, make better decisions because of those measurements.
I founded the first company where you spit into a tube and you sequence the DNA and we say what diseases you'll get. When I did that, Steve Jobs called me up and goes, Agus, you screwed up. I go, what did I do? He goes, you're telling people about 40 diseases at once. The human brain turns off when you give that much information. And people just, their eyes roll back, they look the other direction. We can't understand so much information right now. And so the challenge is going to be putting the information in a way that disease by disease we can understand, put a hierarchy, know how to intervene, know how to change our lives, and be empowered rather than creating a culture of fear.
But one of the things that is going to happen as we shift to more of these proteomic information is that the information is really going to be more actionable. So if I tell you you have a 4% chance of developing lung cancer, is that really going to change your behavior that much? Are you really going to do anything with that? Probably not. On the other hand, if I tell you you have a polyp growing in your colon and go in and if you get it cut out, nothing will happen. If you don't get it cut out, you'll get colon cancer. Well, that's an actionable piece of information. So I think every time you go to get a physical, you'll find more and more potentially actionable things being measured by these methods that we're talking about.
Unfortunately, we have hit the time limit, and I apologize for not being able to take more questions, but I thank you for listening, and I thank you for being on our journey.